produce the pollen tube. For example, if the pollen grains reach the wrong type of stigma. So what do you mean by this wrong type means? Here, they are incompatible. Incompatible means the one which is not belonging to the same species of the plants. So at that time, the stigma will reject the pollen grains. So what do you mean by rejecting the pollen grains? Rejecting the pollen grains means pollen grains does not, the stigma does not allow the pollen grain to germinate and produce the pollen tube. Actually, when the pollen grains reach the stigma, there is an dialogue between the stigma and the pollen grains. So there's a continuous dialogue between. What do you mean by this dialogue means? Actually, there's a chemical interaction. There's a chemical interaction between stigma and the pollen grain. So this chemical interaction is called as dialogue between the pollen grain and the stigma. So what is this chemical interaction means? The stigma will secrete some chemicals. So these chemicals will contain some sort of sugary substances. Sugary substances. These sugary substances will stimulate the pollen grain to germinate. The sugary substances are the chemical. The stigma will secrete the chemical that will stimulate the pollen grain to germinate. So that is taking place in the right type of stigma. That is what we call it as a pollen pistil interaction. So pollen pistil interaction not only includes this step, but further the pollen tube have to pass through the pollen tube that have been formed from the pollen grain. So here pollen tube is nothing but the extension of intine wall of the pollen grain. Intine wall of the pollen grain, and generally through which structure does the pollen tube will form means it is through the germ pore region. So already we had discussed in the earlier classes, germ pore is a region of exine where the sporopollen is not deposited or sporopollen is absent. Through that thin area, the intine wall will extend and form into a pollen tube. And not only this pollen tube that have been coming from the pollen grain, so that pollen tube will pierce through the stigma and through the tissue of style. And whenever it is passing through the style, later it will enters into the ovary and reach up to ovule. So starting from the germination of the pollen tube up to the time where the pollen tube reaches up to the ovule, the entire process is called as a pollen pistil interaction. So this pollen pistil interaction is related with because is concerned with the some sort of chemical stimulus. So here the pollen tube is formed because of the chemical stimulus. So we can call it as a chemotropic growth. So chemotropism also. That is pollen tube is growing through a stimulus called as a chemicals which are secreted from the stigma. That's why we call them as a chemotropism or chemotropic growth. Okay. So let us see how the further process will be carried out. That is called as a fertilization process. So here, before going to the process of fertilization, we should know something about how the pollen tube enters into the ovule. The pollen tube enters into ovule by 3 methods. Okay, those 3 methods are, the first one is called as porogamy. So porogamy means the pollen tube enters into the ovule through micropyle. Pollen tubes enters into ovule through the pore. So that pore is nothing but micropyle. That is from the anterior side. That is from the anterior side. This type of condition can be seen in examples like hibiscus and Otelia plants. And second one is called as chelazogamy. So chelazogamy was discovered by Trube scientist in a plant called as Casuarina plant. So Trube had discovered chelazogamy in a plant called as Trube Casuarina plant. Here, the pollen tube enters into ovule through chelaza. Through chelaza. That is from the posterior side. That is from the posterior side. And the third one is called as mesogamy. So this can be seen in examples like in cucurbita. Cucurbita, where the pollen tube enters into ovule through either funicle or integuments. That is from the sides. So from the lateral sides, it will be entering. So we call this as a mesogamy. So whatever may be the method, the ultimate thing is the pollen tube have to enter into the ovule so that the further process called as fertilization will be taking place. So here, the pollen tube enters into the ovule. After entry of the pollen tube into the ovule, what happens? The pollen tube is directed towards embryo sac. So because we know that inside the ovule, nucellus is present, and in that nucellar tissue, a female gametophyte is present. That female gametophyte is called as a embryo sac. So that is encouraged or promoted by a structure called as filiform apparatus, which is present in the synergids. The pollen tube is directed towards embryo sac by the chemicals secreted by filiform apparatus. So where does this filiform apparatus is present means the filiform apparatus is present in the synergid cells. So in the embryo sac, we know that egg apparatus is present. Egg apparatus contains 3 cells total. One is a large cell called as egg cell, which is present in the center. On either side of the egg cell, 2 small cells are present called as synergids. And on the micropylar side of those synergids, we can see the presence of finger-like projections. So those finger-like projections are called as filiform apparatus. Those structures will secrete a chemical so that the pollen tube is directed towards the embryo sac. Okay. So here pollen tube enters into embryo sac through the one of the synergids. So if the pollen tube entering into the embryo sac through one of the synergids, that synergid will get degenerated. That synergid cell is degenerated, through which the pollen tube is entering into the embryo sac, that synergid cell is degenerated. And once the pollen tube enters into the embryo sac, so the pollen tube now contains total 3 cells. Those 3 cells are a vegetative cell and 2 male gametes. So this structure where a vegetative cell and 2 male gametes are present, that entire structure is called as a male gametophyte. So the male gametophyte contains total 3 cells, whereas the female gametophyte contains 7 cells. Female gametophyte contains 7 cells. So here, the pollen tube will burst into the embryo sac. So whenever it is bursting into the embryo sac, and it will releases the 2 male gametes into the embryo sac. It will releases 2 male gametes into the embryo sac. When these 2 male gametes are released into the embryo sac, so those 2 male gametes will help in a process called as a fertilization process. So here, out of the 2 male gametes, the first male gamete goes towards the egg cell, that is called as a female gamete. That is called as female gamete, and it will fuse with the female gamete, resulting into the formation of a structure called as a zygote. And as we know that male gamete is haploid, which contains one set of chromosomes. Egg cell is also haploid, which also contains one set of chromosomes. And these 2 sets of chromosomes are united to form a diploid structure called as a zygote. And this process is called as first fertilization process. First fertilization, and technically it is called as syngamy. Syngamy process. So where does syngamy is taking place means syngamy is taking place inside the embryo sac by the fusion of one of the male gamete with that of the egg cell. Okay. So this process is called as a syngamy or first fertilization process, where male and female gamete are participating in the process. And this zygote that have been formed here, this zygote is the first cell of male sporophyte generation. It is the first cell of sporophytic generation, a sporophyte stage. So from there, the next generation of the plant body will be produced. That's why we call it as the first cell of the sporophyte stage. And here, the second male gamete is there, no? That second male gamete, which is also haploid, And by the meantime, we know that the embryo sac contains a polar nucleus, or polar nuclei are present, upper polar nucleus and the lower polar nucleus. By the time, what happens? The 2 polar nuclei will fuse and convert it into a structure called as a secondary nucleus. And as the 2 polar nuclei are fused, that is, each of the polar nuclei is haploid structure. So they are converted into a secondary nucleus called as a diploid structure. And this will be resulting into a structure called as a primary endosperm cell. Primary endosperm cell, or otherwise it is called as primary endosperm nucleus also. So here, 3 sets of chromosomes are fused. 1 set of chromosomes from the male gametophyte and 2 sets of chromosomes from the female gametophyte. So total 3 sets are fused. So here the so formed primary endosperm nucleus is an triploid structure. It is an triploid structure. And here, as the 3 sets of chromosomes are fused, we call this process as a triple fusion, or otherwise it is called as second fertilization process. Second fertilization process. And here 2 times of fertilization is taking place. But the difference is, in the first fertilization, both male and female gamete are participating. So we consider this as a true fertilization process. This is considered as true fertilization process. Whereas in the second fertilization, male gamete is participating while the female gamete is not participating. But instead of the female gamete, the cells of female gametophyte are participating here. So that's the reason why it is called as vegetative fertilization process or triple fusion process, as 2 times the fertilization process is taking place. We call this process as the double fertilization. We call this process double fertilization, and double fertilization is a unique feature present in angiosperms. Unique feature of angiosperms. So no other group of plants have this type of characteristic feature. So only in the angiosperms. Okay, if you take the lower other plants like gymnosperms and pteridophytes, bryophytes, in all of them, only one fertilization will be taking place. But here, 2 times of fertilization is taking place. So we call it as a double fertilization process. So this is a way how this process of fertilization will be taking place, leading to the formation of zygote and primary endosperm nucleus. And here, there are some changes which are taking place as a result of post-fertilization process. That is the changes which is taking place after fertilization. So called as a post-fertilization structures and post-fertilization events. So here, what are the changes which are taking place in this process? The parts of a leaf will fall off. For example, the calyx, that is sepals, petals, that is corolla, stamens, that is androecium, stigma, and style of the carpels. All these parts will wither and fall off. All these parts will wither and fall off. Whereas the ovules which are present inside the ovary, they are converted into seeds, and ovary is converted into fruit or fruit wall. Fruit or the fruit wall. And here the parts of the ovule, that is the micropyle which is present on the upper side of the ovule, will remain as a opening of on the seeds. Opening on the seeds. And the funicle of the ovule will remain as a stalk of seed, and the hilum of the ovule is remained as the point of attachment of seed to the fruit. The point of attachment of seed to the fruit. And the zygote that have been formed as a result of fertilization will be undergoing mitosis, leading to the formation of a structure called as embryo, which undergoes mitosis to form into embryo. And the primary endosperm cell or primary endosperm nucleus, that will be converted into endosperm, which is also triploid in its condition. Triploid in its condition. So here, during the formation of endosperm, initially free nuclear division will be taking place. During the formation of primary endosperm cell into the endosperm, free nuclear division will be taking place. That is, already we had discussed about this process in the earlier classes. Like free nuclear division means the nucleus divide continuously. Nucleus divide continuously, so that leading to the formation of multinucleated condition. And later, after some time, the cellularization will be taking place. Cellularization means the cell cytoplasm, each nucleus will forms some accumulate some cytoplasm and also cell wall around it, leading to the formation of cells. So this type of changes will be taking place during the formation of primary endosperm cell into the endosperm. And endosperm is rich in nutrients. It is rich in nutrients, or it contains abundant reserve food materials. abundant reserve food materials are present in it. And what is the use of this abundant reserve food materials means? It is providing nourishment to the developing embryos, to the developing embryo. So that is the significance of the endosperm. And if you could recollect the nature of endosperm present in the Cocos nucifera, in coconut, the liquid endosperm is present. This liquid endosperm is nothing but the coconut milk. And apart from this one, they have the solid endosperm. The solid endosperm is nothing but the kernel, which is the edible part of the coconut. So that is initial the free nuclear division leading to the coconut milk. Gradually, it is converted into solid endosperm in the form of the kernel. That is what initially the free nuclear division will be there, leading to the formation of later into the cellularization process. So here there are two types of seeds are there depending upon the presence and absence of endosperm. Those are called as endospermic seeds and non-endospermic seeds. So by the time of maturity of the seeds, if the endosperm is there in the seeds, such type of seeds are called as endospermic seeds. Okay. Endosperm remains in the matured seeds. Remain in the matured seeds. So that means some amount is utilized for the development of the embryos, but still some amount of endosperm will be remain in the mature seeds. So such type of seeds are called endospermic seeds, which can be seen in the examples like in castor. Whereas, so endosperm is completely utilized by the developing embryos. So that endosperm is not remained in the matured seeds. Such type of seeds are called as non-endospermic seeds, where the developing embryo completely consumes the endosperm. So this type of seeds can be seen in examples like in pea, beans, and groundnut, like examples. So that is the difference that can be seen depending upon the presence and absence of endosperm by the time the seed is undergoing maturity. Okay. Then coming to next structure, that is embryo. So embryo is the structure which is developed from the zygote by a process called as mitosis. Okay. So here embryo is present inside the seed along with endosperm. And we know that every embryo have three parts. So those three parts are radicle, plumule, and cotyledons. So radicle, when the seed germinates, we know that seed forms a small plantlet. Actually, when the seed germinates, which part of the seed is developing into plant means the embryo part. The parts of the embryo will be given rise to the next generation of plants. So radicle will give rise to the root system of the plant. Plumule will give rise to the shoot system of the plants, and the cotyledons will give rise to the primary leaves, the first formal leaves which are providing nutrition to the developing root system. So here the primary leaves are retained till the formation of normal green leaves in the plant. Okay, so radicle will grow towards downside, grow downwards into the soil, and the shoot system will grow upwards above the soil. And which structure will come out first means the radicle will pierce through the root, pierce through the embryo and forms the root system first. Later, the plumule will come out and convert into shoot system. So the shoot will contain the primary leaves, which is providing nutrition to that one. And this entire process of development of embryo, starting from male gamete and female gamete formation, all the stages of embryo development is called as the embryogeny. And in this process, so there are different stages of embryo formation is there. Those are called as, first one is grow embryo. The second stage is called as globular embryo. Third stage is called as heart-shaped embryo. And the completely developed embryo, that is called as mature embryo. So here pro-embryo, actually it is developing from a structure called as zygote. So zygote is a unicellular and a diploid structure. So this will divide to form 2 cells and later into 4 cells, like 2-celled and 4-celled structure, leading to the formation of a stage called as a pro-embryo stage. Pro-embryo stage, and later gradually the mitosis will increases, so that forming into a long and elongated structure with a suspensor like this. So this type of structure with a globular organization. So this globular organization structure is called as a globular embryo. Globular embryo, so which contains a suspensor. And later, gradually, along with the suspensor, the lower globular part will be converted into a heart-shaped structure, like this. Heart-shaped structure. So this is a suspensor. And later, this entire structure will be converted into a matured embryo with developed parts. Like this. So all the cells will be present. Like this. So here, this part is called as radicle. So this is called as suspensor. And here, the plumule will be present. And 2 cotyledons are present. So here, this is a diagram related to the dicot seeds. That's why 2 cotyledons are shown here. So here, these are the different stages of embryo formation, starting from zygote, leading to formation of pro-embryo, from there globular embryo, from there heart-shaped embryo, and forming into a matured embryo. And generally, every seed will have seed coat. So seed coat may be either 1 layer or 2 layered. So depending upon that one. So testa and tegma or tegmen are present. Testa is the outer seed coat, and tegmen is the inner seed coat. Actually, these are nothing but the outer integument of the ovule and inner integument of the ovule. And inside the seed coat, we can see the presence of endosperm and the embryo. Endosperm and the embryo. So they are basically, there's a difference between the dicot seed and the monocot seed. Let us see the diagrammatic representation along with the parts here. So if you see the embryo of dicot plants, so they have big cotyledons, 2 cotyledons are there. So fleshy cotyledons, 2 fleshy cotyledons are present. These fleshy cotyledons will store food materials. Will store the food materials. And the point of this is the embryonal axis, the main body of the embryo, embryonal axis. So here the point where the cotyledon is attached to the embryonal axis, that part is called as cotyledonary node. Cotyledonary node. And the part of the embryonal axis above the cotyledonary node is called as epicotyle. Epicotyle. And the part of the embryo below the cotyledonary node is called as a hypocotyle. And the tip of the hypocotyle is called as coleorhiza or radicle, and the upper part is called as a plumule. Radicle, and this part is called as a plumule. So these are the parts which are present in the dicot seed. Whereas in the monocot seed, here cotyledons are only one cotyledon is present, and it is shield-like. So that shield-like cotyledon is called as scutellum. Shield-like cotyledon is called as a scutellum, and here majority, and of course it does not store the food materials. Whereas in the dicot seed, we said that the cotyledons are storing the food materials. So if you could see the diagram of the monocot seed, the embryo is pushed to one end, to the lower end. So this is the embryo part. Embryo is pushed to one end, and majority part is occupied by the endosperm. And endosperm will store the food materials. Endosperms will store the food materials in it. And separating the endosperm from that of the embryo, there is an triploid membrane is present. That triploid membrane is called as aleurone layer. Aleurone layer. And here in this embryo, the plumule is present, radicle is present, and covering the plumule, there is an covering what you call envelope-like structure is present. That envelope-like structure is called as coleoptile. In the same way, the radicle is also surrounded by a membrane. That membrane is called as coleorhiza. Coleorhiza. So these are the characteristic differences that can be seen between the dicot seed and the monocotyledonous seeds.